problem. I just want to show you just a few slides, and then we'll get into using the programs. On things that AlphaFold can do that maybe you haven't thought about using them for. Just to give you a sense of some potential projects. I'm very interested in kinases. We've done a lot of work on the activation loop of kinases, what makes a kinase active or inactive. But I was talking to Richard Bayless, my friend at the University of Leeds. I go there every 6 months. I have a sort of fellowship to visit there. And we got talking about the domain structure of kinases. So most kinases have other protein domains that help with the function. They help with activating it, inhibiting it, membrane binding, substrate association, all sorts of different things. So here's two examples of PRKD1 and RET, where I've colored every domain a different color. So you can see how complex they are and all of the disorder. So what I did was I used 25 models of all 480 proteins that humans have that have kinase domains. I ranked them, and I opened up every single one in PyMOL and looked at them over several months, all 480. And I wrote down where the domains were. I started with what's called the ECOD database, Evolutionary Classification of Domain, ECOD, University of Texas. So I started with those assignments, but then I modified them, made them a little longer because, the AlphaFold model had an extra helix that is not present in the standard domain definition of that protein. Or there were some small domains that, a little folded bundle that AlphaFold scored pretty well that don't have a name and have never been seen before, and they are novel domains. And so you can make these, we made these domain diagrams for all 480 kinase genes. And we worked on the colors. Actually, we worked on the fonts are much bigger now than they were on this one. But I have these for all 480. So if you're interested in a kinase, if you go, every paper you've ever seen on a protein gives a domain diagram in the first figure. They're often nonsense. They often don't have the boundaries. They're often ugly. They're not to scale. We're doing this for every kinase protein to scale with consistent names across all of the proteins. So I just want to put that out there if you're interested in a kinase. This is one use of AlphaFold. Another thing in AlphaFold that I want to make sure people understand is the ability to get different conformations of complex proteins. And so a lot of people say, AlphaFold only gives you one model. No matter how many times you run it, all look the same. It's not telling you anything. That is definitely not true. If I would always run the program to give you 50 models. Let's say you're working on one protein, give you 50 models of that one protein, and don't just look at rank number one. You want to look at the ensemble of all 50 models, because what you may see is that certain domains within those 50 models interact differently in one subset of the structures than they do in another subset of the structures, and that may be biologically correct. So here's an example. This is a protein called PLC gamma 2 phospholipase. It binds a kinase called BTK and a protein called RAC2. It has many domains. You can see the sort of diagram up here, and it makes this kind of big globular thing. There's this kind of 500 residue inserted region in the middle of a pH domain that is kind of the auto-inhibitory thing. So it almost acts like a lid. You can see the cartoon in the paper about this protein, where this whole lid just comes off like this, and then that exposes the active residues that are the phospholipase activity. But nobody knows the structure of this thing. So we ran AlphaFold lots and lots of times. We did it in the presence of BTK and RAC2, two protein. RAC2 is an activating protein for this function. And if you run it with RAC2, you get this model. And so the parts of it that are the same, like this part is all kind of oriented the same, I think, as it is in this one. Anyway, you can see this whole part of it has come off like a lid. And again, so if I run these 50 times, I get a lot of structures that look kind of, globular, the whole big thing. And then I get a set of structures that are open. And this helix is formed that is not present here. It's kind of like a spring. This helix forms and it moves the entire regulatory domain off of the catalytic sites to expose the protein. So, even on a single protein, this is telling you something about the biology of how this protein works. A second example is JAK1. I ran lots of calculations with JAK1 for Gareth Thomas, one of my colleagues, and I got 3 different conformations of just monomeric JAK1. So JAK1 has a firm domain, SH2, a pseudokinase, and a kinase. And basically, this part was all the same, from firm all the way through pseudokinase, but the kinase was in 3 different positions. I didn't know what it was. One of them was like an auto-inhibited structure of type 2, related protein. But then this cryo-EM paper came out of a dimeric homodimer of JAK1, maybe JAK2, And it looks like this, where the kinase domain in some of the structures is outwards like this. And in some of the other cryo-EM structures, it's inward like this. And then there's this auto-inhibited form where the kinase and pseudokinase are kind of facing each other. And then what we think is going on is this is the form that is the kinases are phosphorylating other substrates, other proteins. And this is the autophosphorylation form where one monomer is autophosphorylating the activation loop of the other one. But AlphaFold was able to make all three of these conformations. I just didn't know what they were until this cryo-EM structure came out. I mean, experimental biology is still important to tell us what the models are, mean. But if I had more sense to make the dimer model, I would have noticed this autophosphorylation structure because we looked at those and we said, oh, that's what it is. It's auto. So the lesson is multiple conformations can exist, and you should look at them and not just dismiss them. Okay, this is a summary of everything I've told you. I'm not going to go through it. These are a bunch of the people in the lab right now that are working on kinases and all the AlphaFold stuff we do. Particularly, Qifeng Xu is in my molecular modeling facility and is doing this kind of modeling for anybody at Fox Chase. And there's the funding. Okay, so now we're getting on to the fun part. And so we'll try to finish that within an hour. We might go a little bit. I think the schedule is to finish at 4:30, but we might go maybe 15 minutes more than that. I think you were told it was going to end at 5, so I'm letting you out early. I should, well, maybe. We'll see. I shouldn't have told you that. So, all right. So there's this web page. So it's just my surname, .fccc, Fox Chase Cancer Center, .edu/bioinfo. And there's an opening page there. It has links to these slides, both in PowerPoint and PDF. The PowerPoint is 150 megabytes, but the PDF's smaller. But the PDF doesn't have all the funny animations. So please feel free to download those. There's another on that page. There's also links to a Fox Chase presentation that I did. That has a lot, and I did it for all the postdocs, all the grad students, undergraduates. So it kind of explains a lot about protein structure, hydrophobic effect, hydrogen bonds, how proteins fold, side chain rotamers, kind of really basic structural biology stuff that if you haven't spent a lot of time thinking about protein structure, especially if you're a faculty member over a certain age, you may not have ever really had a course in structural biology. I hope that has changed. I hope graduate students now in biological sciences get a protein structure course, or at least half a course, where they really looked, opened up PyMOL or Chimera, some visualization program, and looked at structures and know what a salt bridge is and know what a disulfide bond is, and all the kinds of details of structure. So that's in at the bottom of that page. There's a set of slides. The other things are just a bunch of links to some of the websites that we're gonna look at now. Okay.
Excuse me.
Yeah.
So when it comes to the IBES score or the IDES score, what numbers are good? Like, what numbers would you consider to be a good starting point for?
Yeah, for the IPSET, for my score, the IPSET score. We are doing a lot more extensive benchmarking than is in the preprint. The preprint is now 1.5 years old, getting pretty well used and cited, which is nice. And so there's a data set in there that says all of the false interactors are well below 0.2, and almost all the true interactors are above 0.2. I would say that a score above 0.4 is probably a correct interaction, and the model is probably close to correct. And you can, those proteins probably interact in that way. Between 0.2 and 0.4, I would say maybe 50% of the time the model is correct. But you may get some false models that will score 0.2, 0.3. Below 0.2, AlphaFold is not giving you any information about whether or how the two proteins interact. If you get a non-zero score, even if it's 0.1, I would look at it, but I'd have to be really desperate I really want to know how those two proteins interact. So I get this really poor scoring model. I would then, if I get a non-zero score, I would look at that. I would look at it very carefully and say, okay, I'm going to run 100 models or 500 models, or I'm going to run different constructs, different sequence lengths. If I'm doing AlphaFold 3, this is a very important feature. It's in the slide later, but I'll say it now. AlphaFold 3 is very sensitive to whether there are biological ligands bound or not. So I did RAS, which binds GTP to a GAP protein, a GTPase activating protein. With GTP, the scores between the two proteins were great. Without GTP, the IPSA and IPTM scores were 0.1. And so it was very sensitive to the presence of a biological ligand GTP. I think the same thing would be true of proteins bound to DNA. Proteins may be bound to certain ligands, drug ligands, probably not so much. Ions, if there is zinc and magnesium ions on the AlphaFold server, you can throw in, got a huge protein, you can throw in 10 magnesium ions, and a couple of them might bind to, or if you have a zinc binding domain, you want to add those things to add context, and those will change the scores. So AlphaFold 3 is very sensitive to that, so let's be careful about it. Okay. I will do a little bit of PyMOL. I think there isn't so much. These are actually ones I was looking at with people here just today. Okay. This is a good example because I'm going to show the predictions from the different output programs on this particular complex. So on the in the slides, there is how to install PyMOL from the open source version of PyMOL, which, trust me, is better than the Schrodinger version, having to do with how you click and how you use the windows. So I would use this version. I'm not going to read this, but you can. It's in the slides and it's on the it's on the web. There's a web page that is how to install PyMOL, and the instructions are there for both Windows and Mac. These are the instructions for Mac. I don't expect you to do it now. There's probably no reason to do that. So want to install PyMOL. There's another program called Chimera X, CHIMERA-X from UCSF. Really powerful. It does a lot more things than PyMOL does, and actually has a really good interface with AlphaFold models. It can read in a PAE matrix, and you can select part of a PAE matrix, and it will show you on the protein where that part is. So you could look at one of those off-diagonal rectangles and figure out, oh, okay, now highlight those on the structure. So it has been, they've added to it the ability to interpret AlphaFold 2 and 3. I think it's both 2 and 3 interactions. So Chimera X is really powerful. I don't like the pictures of proteins it makes. It makes them look like ribbon candy. They look like you just want to eat them and get a cavity or something, and become diabetic maybe. But you can change those settings so that you can make it better looking. I like the images that PyMOL produces. PyMOL is also smaller and simpler, so it's kind of easier to get a hold of, hang on, than Chimera. So I typically use PyMOL. People in my lab, some of them use Chimera, and we have fights about it, like which one's better. Okay, so you can all go to the PDB, Protein Data Bank. The link is in that set of links on that bioinfo page. And you can download the structure of a protein. So I can search for P53. And I get a whole lot of structures. Each of these has a PDB code. It begins with a number and 3 letters or numbers following it. The very sad thing is, as of next summer, they are going to run out of 4-character PDB codes. And they are going to do this monstrosity, which is PDB underscore and 8 characters. Old PDB will be the 4 will be just the last 4 characters, but then they add these unnecessary zeros. But they will start using 5-character PDB codes next summer. I think I should retire before that. Absolutely. But there's this PDB code. So I can go to PyMOL. So let this is 8 UQS came up. And I can just type the line fetch 8, what did I say? UQS? I don't know, it doesn't matter. And hit return. And it goes and downloads it. So there's 8 UQS. And so any experimental structure is in the PDB. You can immediately go to PyMOL, open it up. If you put PyMOL on your Mac, you can go to a terminal window. So this is the terminal program. It's up, and these are all of the. Oops. So the terminal program is one of the utilities that's installed on your Mac. Windows has something similar to it. But in one of those, you can just say, once I've installed PyMOL, I can just type PyMOL. And I use an ampersand to make it in the background, and a PyMOL window comes up. And now I can just say fetch my favorite PDB structure, 1OL5. And there it is. And I can start looking at it. You can do the same thing for if you have you run AlphaFold and you have a folder that has 5 models in it. Let's see if I can find one. This is a set that actually Ben asked people if they wanted to submit sets of structures. PyMOL, AlphaFold sets. This is one of those sets. And so there are CIF files. The extension is .cif. That stands for crystallographic information format or file. It's the modern format for a PDB structure, for an experimental structure or predicted structure of a protein, the coordinate, XYZ coordinates of all of the atoms. And it has information about the sequence and the database and the authors and the crystallization conditions and all sorts of information. There is an older format that ends .pdb, Protein Data Bank, that goes back to the 1970s or even 60s. And the lines are all 80 characters long. And because I'm not old enough to have used them, but I'm old enough to have seen them, you get these boxes of these punch cards. And that's how you fed information into a program into a computer or data into a computer, was these punch cards. And they stack them up. And each one had 80 columns and all the letters and the numbers for 80 columns. And so it's a very old format. And it's all kind of scrunched together. Things, there's only 4 columns for the residue number. You can't have a number bigger than that. So the MMCIF format is the modern format, and you should really use it and not use the old PDB format. Just a soapbox. I'm on the PDB's advisory board, so I get to work with them and tell them what to do sometimes. I'm in this particular folder. And there are 5 different models. They all begin fold, MDC, AD, long name. But I can do something like PyMOL, type the word, fold, star, CIF. So I want to get the star as just a wild character. So anything that begins with fold and ends with CIF, and I just type the word PyMOL in front of it, and those all pop up all at once. And I can begin to look at them. So over here, there's a window that lists all of the different structures that are live inside the file. And I can turn them all off, except the first one. So now I'm just looking at the first one. And I can look at each one separately. I'm going to go back to the one that I was looking at before. This is a predicted structure of KRAS, which is the magenta protein. This is a Boltz-2 structure prediction of KRAS and magenta and the RAS binding domain of RAF1. I've already shown you RAF1 with the kinase domain and the CRD domain and the binding to KSR1. That was my IPSA example. This is just the RAS binding domain of RAF1. And it makes this sort of complex. The default when it opens is this kind of cartoon diagram. It's called cartoon by PyMOL. That if you want to hide cartoon, you can hide cartoon. That's this Richardson diagram that comes from Jane Richardson. And so that's one representation of the structure. Where the helices, I'm sure you've all seen these helices are, like this, and the beta sheets have arrows, that kind of, it's called a Richardson diagram. But I can represent the structure in all sorts of different ways. So if I go over here, here's the name of the structure. So this was the name of the PDB file or MMCIF file. And I can click it on and off. But I can, the buttons here are A, S, H, L, and C. What is S? S is show. So I can show spheres. And so now for every atom, I have a sphere of about the right size. The H is for hide, not surprisingly. And so I can hide all the spheres. And I get rid of them. I can show, this, what's usually very interesting is to show the surface. So if I were a little ant or small creature inside of a cell and approaching this is roughly what I would see. Not in color, but I don't know. That's the kind of van der Waals surface of the protein. So we can hide that. we can, if we hit the button down here that has an S on it, that's for sequence. And now I can see the sequence up here. One of the things with the sequence is I can select a piece of the sequence. So I'm just going to click on the sequence and drag my mouse. And now I have selected a portion of the sequence of one of the two proteins. And it's highlighted with little dots, that section. And you can see over here, this SELE thing has popped up. That is now an object inside PyMOL that is just the thing, just the part that I've selected. And so all these A, S, H, L, and C all now are just on that selection piece, not on everything else. So I can show just the spheres of that segment or hide them. The C is for color. So I can choose color. I can make that segment yellow, and I get 6 different kinds of yellow. I'm just going to go with plain yellow, yellow. And so now that section is yellow. Very useful for, isolating certain parts of the structure, coloring them different, representing them differently, whatever. I can, let's see. If I apply it, so you've got all sorts of different colors. You see this one where so I've hit the C button next to the bolts, the whole model. And I've said, hit the C, and there's a thing called spectrum. And it says by B factors, the next menu. This is a model from Boltz-2. So what's in the B factor field is the PLDDT of that amino acid. Now we all know what PLDDT is and how it's calculated. So this structure is now colored by the PLDDT. And you can see that the C terminus is a little bit, and it's this particular coloring scheme. Red is high PLDDT, blue is very poor PLDDT. There are other commands you can give to use different coloring schemes if you want. But you can see, so part of the RAS binding domain is, it's not so well predicted. Maybe that's a little less stable, or, it's on the surface. So you can color by PLDDT. But then you can also go back and color by chain. And it's just by chain. And now we're back to coloring it by chain, where chain A is green and chain B is blue. What else? There is the action. So A is for action. You can center it by just hitting the orient button. Sometimes that's useful if you're trying to move it around and it's not centered. It's a problem. Moving it is just, click on the field and just move your mouse, and you can rotate in any direction. I like to use under the mouse. Up here, you can set how the mouse functions. I find one button viewing to be the easiest. And down here is what one button viewing does. So for just the regular left mouse, it just rotates. If I click, I just rotate. But if I use the command key, I can now zoom in and out. If I use on a Mac, if I use the option key, I can move left and right. So it's very useful for kind of moving things around. Same thing exists in the Windows version, and if you have a mouse with 3 buttons, the buttons will behave differently. You can experiment with how to move it using those different kinds of commands. I can, if I hit the command key, and swipe my finger on the trackpad, it zooms in, and I can, zoom in on any part of the structure. So I'm going to click on the GTP, which is bound to the RAS. And so that's now the selection object. One thing that is very useful for figuring out how 2 molecules interact is over here on this. So I select something. I could select the whole chain, or I could just select the ligand. Under the A menu, if I scroll down, there's something called modify. So basically, it's going to modify the selection. How do we want to modify it? We want to modify it around all the residues within 5 angstroms. So I'm going to turn the selection into, it was just GTP. Now I want it to give me all of the full residues that any of the atoms are within 5 angstroms of the GTP. I always use 5. Hydrogen bonds are 3.5 angstroms. Most hydrophobic contacts are 5 or less. So anything over 5 is not really quite in contact. So for atoms contacting each other between residues or between proteins, you look at everything 5 or less. You can do 6 if you want, but anything much bigger than that is really not a contact. There are plenty of contacts between 4 and 5 angstroms, so don't use 4. So it gives me this option of residues within 5, and if I just click that, now I get a big selection. And those are all the residues around it that are within 5 angstrom. Now the selection button works on those. So I could say show the sticks. I could say color orange. I could say label, L is for label. I could label the residues with a 1-letter code. So now I have, and I'm going to do one more thing for this selection. Under the color, I'm going to say color it by element. And watch what happens. So if I color by element, nitrogens are blue because the sky is blue. Oxygens are red because oxygen's fire and red, and your blood is red with oxygen in it. And sulfur is yellow because sulfur is yellow. And the carbons all stay whatever the color was before I did that command. This is very helpful because we can zoom in on that GTP and look at all the amino acids that are really nearby. And are there any favorable interactions maybe between the phosphate and residues in the protein? And of course, it's going to be harder to find them when I have to do this live. Well, there's at least one. There is so up here, there's a whole bunch of menus. You can modify the display in all sorts of interesting ways. You can set things like the sphere radius if you want to make the spheres really small. All sorts of commands that you can specify to modify what you're looking at here. But one very useful thing that we haven't looked at is the wizard. And under the wizard, there are different wizards. One of them is measurement. The second one here says measurement. So if I click that, now you can see in the upper left, it says, please click on the first atom, very polite. And down here, it says measuring distances, which is what we're going to do here. And it says, click on the first atom. So I'm going to click on one of the atoms of the GTP, this oxygen. And you can see it's highlighted with a little square on it. Now on the upper left, it says, click on the second atom. And I can see, just because I'm used to looking at these things, there's this blue nitrogen that's kind of pointed right at that oxygen. So I'm going to click that blue nitrogen of the backbone. And then the distance appears and a little dotted line. So there's a 2.7 angstrom distance between that oxygen of GTP and that NH of the backbone of residue, whatever it is, 25. No, sorry, it says alanine 18. And that's what you're looking for. When you look at interaction between two proteins or a small molecule and a protein, you want to look at a lot of the hydrogen bonding interactions. So those are all the ones that are about 3 angstroms, which is about 2 covalent bonds. So that's why I know how far to look is if I look at this side chain right here, that's 1 covalent, that's 1 carbon, that's 1 carbon. That scale is 1 covalent bond. The interaction between a nitrogen and an oxygen that's forming a hydrogen bond with a NH and an O is slightly positive H on the NH and slightly negative oxygen on the O charges need to be about 2 chemical bonds apart. So that gives you kind of a frame of reference for kind of identifying where the contacts are, and you want to look at all the hydrogen bonds. There are ways in PyMOL to do a selection and have it find all the hydrogen bonds and all the polar contacts. I'm not going to do that because I never use it. I always do it by eye because I'm used to it. But there are ways to do it sort of more automatically. So I would take this GTP, and nothing beats looking at the structure. I have to say this to my students all the time because they will send me huge amounts of data. I'm like, well, did you look at the models in PyMOL? No. I looked at the models in PyMOL, and those proteins are not interacting correctly, or it's just spaghetti, or it isn't saying what you're measuring. You measure the distance between this and this. I looked at it in the structure, and that's not the distance at all. You have a bug somewhere. Totally fine, totally reasonable, happens all the time. But you need to look at the structure, and it needs to agree with what the PAE diagrams are telling you, or what the AlphaFold scores are telling you, or what you know about how the proteins interact. You want to see it inside the structure. Can't emphasize that enough. And so you can poke around and basically click on a lot of things. Let me do it in this one. This is also the one that got sent to us. So this is an AlphaFold model of two proteins. Let me just color them by the two different things. So I have a little alias that colors by chain. So one protein is magenta, one protein is blue. Well, the AlphaFold models initially were all over the place, like they weren't all aligned to each other. So I want to do something called align. So I'm going to highlight all the proteins, and I'm going to give a command, which I should have already done here. Oops. Maybe it's down here. Yep. So I know this is hard to read. But down here it says the word align to, ALIGNTO, one word. And I have told it to align to, I started typing the word fold, I hit tab, it completed the full length of this thing. So it's the first object listed here. Fold GLC7_1_431_model_0. So I'm going to align to that whole thing. And I want it to align to that structure, but only chain A, just the magenta protein. So I'm going to align all 5 of these models to just chain A of just one protein. And then I hit return, and I've already done it. In fact, we can do it for this one where I haven't already done it, just to show how magic it is. So these are not aligned, right? And you wouldn't be able to tell very quickly whether these models are the same as each other or different if you don't align them. So I'm going to pick a segment. So this long helix looks like it's pretty good segment. If I click on it, up here it tells me, oh, it's chain A and it's residue 188. And the other end of it, it's chain A and it's residue 298. So I can go up to the top. I can click at the top or down at the bottom, and I can say align to, ALIGNTO, space. Start typing fold, hit the tab key, and it finished, completes it. And here it tells you, oh, there's everything, that whole string, and 0, 1, 2, 3, or 4. So I'm going to just pick 0. And residues, whatever I just said, 188 to 298. Need the word RESI, residue. And watch what happens. Like magic. And so you can do that with any selection.
You can just do it if you had something selected, you can just say and SEL again.
Yes, if you already have something selected, you could say SELE space SELE, which is what you currently have selected, and chain A, and resi something to something, anything you want. And so for AlphaFold, you definitely want to do this, and you want to pick the folded part of one chain of one of the models. All of the things have to be highlighted for you to be able to if only 2 of them. So if I give that command again, it's only going to align the 2 that are lit up over here. So it's only going to give me the RMSD and it gives you the RMSD. Remember RMSD in angstroms and how many residues it calculated it over. Of the 2 that I just aligned. So now you can begin to look at this and say, oh, there's a variation in this helix. If I align on that part of the structure, this little mini domain in the blue protein is moving a little bit. There's uncertainty in it. And so you want to be able to look at all of your structures in a common reference frame. So again, it's aligning on one thing and looking at everything else, investigating everything else. The scores work the same way. Your brain visualizing things also works this way. I think that's about all I want to say on PyMOL. I know that's a whirlwind tour. On the YouTube channel, there is this 2-hour lecture beginning of the pandemic that was 2 hours about how to use PyMOL. So all of these things plus a whole lot more. So, okay. Now let's do just a little bit of AlphaFold for the next half hour or so, which I think is enough time to just do a few things. So let's go back to the slides. Remember Command L magically appears. All right, so that's installing it. Here's a list of all of those commands. I think I've told you most. You can save a PyMOL session. I haven't told you that. I did the selection stuff, aligning images. There's a button in the upper right where you can make a PNG file, PNG image of exactly what you see on the screen. So it's super. Okay. So we're going to really look at AlphaFold 2, AlphaFold 3, and Boltz-2. Those are the most commonly used programs really right now. There is also OpenFold 3, which is this consortium of both academic and industry people who are getting together to get a lot more protein ligand data from these companies to train new models that maybe do a better job with small molecule ligands. So there's a lot of excitement around that. One of my former students is one of the organizers. So keep your eye out. There's also Chai1, CHAI1, and maybe there's a Chai2 now. So there are like all sorts of rival programs that are like AlphaFold 2 and 3 that are doing some things, and maybe some of them have different advantages and disadvantages. You might want to run each of them on your system to see if something different pops up that might be intriguing. So these are the ways to run these three programs. AlphaFold 2, the way that most people will do it is the ColabFold Jupyter Notebook. A Jupyter Notebook is just a web page that has a program in it, and all of the lines of the program you can execute like certain parts of a program. Collab means Google Colab. So Google, which sometimes we think of as the evil empire, among many evil empires. One thing that they do that is a service to the community is they have something called Collab, which is a person can set up a Jupyter Notebook that needs GPUs. So these programs all run in graphical processing units, GPUs. They need to have your computer needs to have a GPU in it. You wouldn't want to run any of these programs on your Mac. You would run them on a Linux machine that has a GPU in it. I don't know about running it within Windows. I'd be too scared to try, but probably you can do it in Windows. But the Mac GPU is not the right kind of GPU. So there's certain kinds of GPUs, most of which are made by the company NVIDIA. So what Collab has done is you're basically, you click, you say run this program and do this calculation. And you're using GPUs owned by Google. And they are running, because they've got excess capacity because they've got, extremely large resources. And so you're using their GPU time. If you spend $10 a month for a Collab account, you get more time and more, it doesn't time out on you. You can buy a certain number of credits. There are different ways to do that. So you can run AlphaFold 2. ColabFold is only AlphaFold 2. This ColabFold webpage, which is put up by Sergey Ovchinnikov. That's SL Krypton here. So if you just Google ColabFold, you'll find it. But that's the webpage. You can run ColabFold on your own local machines, just as a command line program. If you know how to run a GPU, and if maybe on the cluster HPC computers here, is it probably all set up? It's probably all set up for you in a way that you could use locally here in Utah. There is the AlphaFold 3 server, which is that address. Has limited capability, but there are interesting things you could do on it. You can run AlphaFold 3 now. Well, you could 6 months after the paper came out. You can run AlphaFold 3 on your own machines, again on a GPU cluster. There are several Boltz-2 servers, several places where you can get Boltz-2. Down here, I've just put a sort of trial sequence that we might use. So, what is that? This picture can't be displayed. I don't even know what it was. Anyway, doesn't matter. So let's go through ColabFold first. So here are a lot of text hints and things to do, but it's probably better to just look at the web page. Can I find it really quickly? Oh, it's hiding the windows. Okay. There's the AlphaFold server. There it is. My runtime is disconnected. Close. Okay, so this is what the ColabFold web page looks like. The guy that runs this likes these little cartoons. He has a different cartoon animal for every server that he has. And I can make this bigger. So here's all the specificity. We're on 1.6.1 now. So, what does AlphaFold need? It needs a sequence, one or more sequences, and this is AlphaFold 2, so only proteins, no DNA, RNA, post-translational modification. Only a protein sequence or more, and some parameters to tell it how to run, whether you use templates or not. So there are all these. One of the wonderful things was, because AlphaFold 2 was released the day the code was released, people could create servers that allowed more accessibility to all the different things that you could set that would make the calculation run differently. And so that's what Sergey and Martin Steinegger did, and Milo Mirdita, is they produced this wonderful, it's amazing how effective and important this was in bringing AlphaFold 2. If everybody had to download it and put it on a GPU machine, very limited numbers of people know how to do that. But there's a web page and you can just put the sequence on and get the results, and and somebody else's GPU. It's great. You have to have a Gmail account to make it run because it's Google. But you put the sequence in here, so the query sequence, and it's very hard to see, but there's a colon right there. And so this is two proteins. This is KRAS and RAF1 RBD. So I've already copied and pasted those into this line. And so the end of one sequence is where that colon is and the beginning of the next sequence. If you want to do a homodimer, you just put in the same sequence twice. Put in the sequence, type a colon, paste the sequence a second time. And you can do multiple proteins, each one separated by a colon. It's very easy to fill out this form. Give the job a name. You want to do that because the PDB files will all have that name in it. And if 6 months from now you want to know where is my KRAS model, it better be in the file name. And if it isn't, you're not going to find it. Otherwise, you'll get some random number attached to it. So put in a job name and I often put in, what residue number did I start at? Because I might do only a piece of the protein, like the C-terminal domain. And I want to know, well, what residue was the first residue? Because when you get the models, that's going to be residue number 1, because that's what you gave it. It doesn't know that it's residue 500 when you do a protein. AlphaFold 2, sometimes when you just make the models, there could be little steric overlaps, or some of the side chain rotamers are not, great dihedral angles. It runs AMBER, which is a molecular dynamics program, to do a little bit of relaxation of the model. And so that's what this numRelax is. You can choose 0, 1, or 5. So you can choose the number of seeds. You could do 16 seeds, 5 models each. You would get 80 models. It's only going to relax the first 1 or the first 5. It won't do all of them, just for CPU reason. So we could set that to 1.
What does relaxation mean?
So in molecular dynamics package is an energy function that makes sure that atoms don't overlap, or that dihedral angles and bond angles are what they should be in bond lengths. So the relax just takes the AlphaFold model and applies that energy function and just does an energy minimization, so that atoms that were too close together really in the model will now come apart to a good distance, and they won't be overlapping. And so the relax is important. Sometimes a hydrogen bond that isn't in the initial model will form after you relax it. Like the distance will be good enough. You can choose templates. If you choose PDB 100, you're getting the whole PDB. So any structure that is in today's PDB could be used as a template to do your modeling. You can also upload a specific template if you want, like a particular, let's say a kinase in a particular conformational state. And maybe you want your other kinase to model, and maybe you want it to look like that state. And if you add just that one structure, it might help. It might not, but it might. I usually leave it at none. I usually kind of want to know what AlphaFold thinks by itself, not what some structure or set of structures. It only uses 4. It finds the closest 4 templates. It only uses 4 of them. I think the output does tell you which 4 it used, but you can't control it unless you choose custom and then tell it which ones that you want to use. I ran this last night on KRAS and RAF1. So there's the sequence. Here are the different other things that you can set. You can set the MSA mode. It's basically how does it produce the multiple sequence alignment. You don't really have to change this. It's just what database of protein sequences was used to make the multiple sequence alignment. Uniref is the UniProt webpage. ENV means environmental sequences, so sequences from soil and ocean, that are not very well characterized. If I had to choose, I would choose just Uniref. If you're working on some weird protein, like some weird bacterial protein that maybe the environmental sequences might help, you can do that. It probably won't change anything from this thing. Pair mode I would leave as unpaired paired. All of these instructions are on the slide. You don't have to memorize them. I just want to show you them and explain them. This is important. So it says model type here, and it says AlphaFold 2 Multimer V3. So that's 2.3 Multimer. One of the choices, well, first of all, if there's an auto, and if you give it a single protein, just a one protein sequence, you're going to get the original AlphaFold 2 from 2021. It is not the one, it is not Multimer that was trained on a later PDB. It was trained on the 2018 PDB, not the 2021 PDB. And lots of extra structures were done in those 3 years. Even if you're doing a single protein, I would manually go in here and select Multimer V3. You can also choose DeepFold V1. I don't know what that is. It's another version. So the auto for single proteins will give you original, well, the PTM version, but you just always want to set it to Multimer V3. The number of recycles, remember I showed you how the data can get recycled, or information out of one part of the network can get recycled to earlier layers. For protein interactions, even for single proteins, I would set this to 6. You can see you run it on your own machine, you can see, and actually on here, you can see after every recycle, you get an IPTM of your 2 proteins and a PTM. And you can see it go up over the first few iterations. Usually around 5 or 6, it plateaus. But the first or second iteration won't be good for a protein-protein interaction. So, and even after 3, it goes up. I would set it to 6. It's going to take more. It's going to take more computer time, more time, but you'll get better looking models. So set that to 6. Recycle early stop tolerance means, well, after every recycle, if the model is within an angstrom RMSD of the last model, maybe there's no reason to keep recycling. So you could save GPU time if you set that. So it's auto. It's the tolerance is 0.5 angstrom. I often just set it to 1, but it doesn't really matter. This is about controlling the relax on Amber. I would just use the default here. The pairing strategy, there are different ways to pair sequences. So AlphaFold 2 Multimer and AlphaFold 3 uses what are called paired sequence alignments. So you've got 2 proteins and you search for all the proteins that are related to those 2 proteins. But there's a protein in mouse related to this protein and a protein in mouse related to this one, like the closest ones in mice. Then the 2 closest sequences to your targets in cows, and then the 2 closest sequence in elephants, the 2 closest sequences in pandas. And now you've got the sequence alignment where it's all on, the 2 proteins are all on one line, and all the different species of the different lines. And what you can get out of that, what AlphaFold is getting out of that, is covariation at different sites. What does covariation mean? It means that either within a protein or even between 2 proteins, you might see big hydrophobic in this set of organisms and small hydrophobic over here. So this is one column, this is another column. There's always big, small. But then in another collection of organisms, maybe insects, it's small and big. But when it's small over here, it's always big over here. When it's big over here, it's always small over here. So they're co-varying. They're varying in the same way. Same thing about charge. You might get positive charge over here, negative charge over here in one set of species, but in another set, you get negative and positive. And that gives you the idea that, well, those 2 residues probably interact because otherwise that covariation wouldn't occur. And so that information in that multiple sequence alignment is used by the networks to kind of figure out proximity of amino acids. It's part of the reason why this works. So that's when it says paired sequences, that's what it means is for paired, if you're doing 2 proteins, it's pairing them up by species. And then the sequence alignment will also have a lot of individual sequences for each of the 2 proteins separately, not paired. You can set the number of seeds. So remember I said do 10 seeds and 5 models each and get 50 models. So you can set this to 1, 2, 4, 8, or 16. Probably for most purposes, I think I would set it to 8. 4 is maybe a little low, but 16 is probably more than you need. But I wouldn't do just 1. I mean, maybe trial calculation, you might just do 1 just to see what it does. But for the real data that you might put in a paper or the model that you might really start planning experiments on, I would do, 50 models or 100 models. So do 8, you'll get 40 models. That's fine. So let's say we would set it to 8. You can control the depth of the sequence alignment. If you want to get more variation in the structures, maybe more conformational change, maybe the relationship between two different domains, the way I showed with Jack1 and PLC Gamma 2, you can set this to lower values. So if I set it to 64, there's two parts of the sequence alignment. So the first part, second part. So there's two numbers. That's why there's two numbers here. But if I set it to a smaller pair, 64, 128, I might see more variation in the models that I get out of the program than if I set it to the default, which is 512 and 1024. So if you're really, you're not getting what you want, or maybe there's some structural variation that you're interested in that you're not seeing in your particular protein, you can set this to lower numbers. This is just about saving. You don't want to hit save all. You'll get these gigabytes worth of files. You really don't need the save recycles means saving the structure after every recycle. You don't necessarily need all of that. I've almost never used that. And that's it. And so once you put in all that information, I mean, the defaults are fine for most things, and really just put in the sequence and name it and then run it. But you could set any of those things. You hit this button up here that's in run all. And you can see this little, at each step of this, there's code underneath each of these steps, under each of these little paragraphs. You'll see this thing kind of spinning around and it's working on your calculation. And you'll see it do one after the other. I won't do it now, as I've already run this. But then it might take 10 minutes for a small protein system. Might take, if you do the Google, if you pay like nobody wants to pay Google more money than it already has, but if you pay them $10 a month, it won't time out. If you're not on this website or on your browser and moving things around, it might time out and you won't get any results. That if you don't pay anything. If you pay something, it won't do that. So actually for this meeting, for this workshop, I paid $10 so that this would work for me today. So it runs. Started at 6:06 and it finished. Actually, it finished at 7:37. So no, it finished at 6:07. So it only took a couple minutes. This is just the sequence alignment stuff. This is the sequence coverage. It tells you, oh, for protein 1. So these are the paired proteins up at the top. So these are homologs of KRAS. These are homologs of RAF1. Then there's a set of sequences of just more homologs of KRAS, but not aligned to anything in RAF1. And then just sequences of RAF1. And the colors tell you the sequence identity. So you can get an idea of the sequence alignment that went into making the model. And maybe that'll tell you something interesting. Maybe it's very shallow and maybe that's why you're not getting good models or good scores. It runs, it did told it to do 3 recycles. There's recycle 0, recycle 1. Here's the IPTM, went from 0.829 to 0.873. And it hit that stop tolerance. And so it stopped after 1 recycle. Here are the, gives you images of the proteins. Here's the colored by chain. Here's colored by PLDDT. There's that tail. And it does this for all, and it gives you the IPTM and the PTM for all 5. So it's making 5 models. I only did 1 seed, I think. So it's giving me 5 models. And so there's model 0, I think they call it, model 2, model 3, model 4, and model 5. And yeah, that's it. And then it ranks them. There's a formula for ranking complexes. It's 80.8 times the IPTM plus 0.2 times the PTM. So the PTM, as I showed earlier, is the whole structure. If there's a lot of disorder, the PTM doesn't necessarily mean very much. But that's the formula for ranking. So these are ranked, and the file names that you get in the output, they'll be rank 001, 002, 3, 4, and 5. And they were for different. The models ran. Remember, I said AlphaFold 2 is 5 sets of weights. That's this where it says model 1, model 4, model 3, model 5, model 2. Those are those 5 sets of weights. But they didn't come out in that order. They came out in a different order in terms of the ranking. So the file names have both this ranking number and this which set of weights produced that particular model. And sometimes when we run 50 models, like only model 4 is producing good complexes of 2 proteins. For some reason, the training went well for your particular target on one of the 5 models. And then you can see the structure. Here are the PAE plots for the 5 models. So I've shown you those. Here's protein 1, here's protein 2. Lots of blue in the off-diagonal rectangles because these proteins, they're crystal structures and they interact really well. And they all look the same, all 5 models turned out the same. It can happen that, yeah, the first 2 have really nice blue off-diagonal rectangles, but the last 2 or 3 don't. So you always need to look at just like looking at the structure, you need to look at the PAE plots. You can calculate all the scores you want. I still want to see the PAE plot. And most reviewers or journals now are requiring PAE plots to be submitted with your models. And that's it. So you get this zip. Oops. Yeah. Okay. So you get a file that has a title like this. The title the name I gave it, which was kras_raf1, the 5-digit number .result.zip. So you want to unzip that. So on a Mac and Windows machine, you just double click it and you get a folder with the same name attached to it. And so I can go into that folder. Oops. No, apparently I don't have it right here. So I'm going to unzip it. And I just change to that directory. It's bigger. And I can look at all the files. Oh, it's in the subfolder. These are all the files that I get. These are the ones that have, and the .pdb, those are the coordinates of the model. They use the old, AlphaFold 2 uses the old PDB format. It has the name, the rank, the model that it came from. In fact, that it was AlphaFold Multimer v3 is in the name. I didn't relax them, so they're all labeled as unrelaxed. You get the figures. So if I just open the PNG files in this folder, the ColabFold output has all of the figures that were on that webpage. So you don't need to download them. They come with the zip file. That's not true of AlphaFold 3, which is really annoying. So for each model, there is also this kind of file, which is kras_raf1 40, all the same name, but at the end of it, it says .json. And a .json, a JSON file, is a text file that has all of the scoring information. It has the PAE matrix values. It has the PLDDT values. So I can just show you one of those. That's just the overall one, but so it says predicted aligned error, and it's got 2 sets of square brackets. It's basically one bracket at the beginning, one bracket at the end, and then let's say there's 200 residues in the structure. There's 200 residues in one square bracket, comma, another 200 numbers in a square bracket, comma. That's each row of the PAE matrix. And so my IPSE score reads this file and gets all the information it needs to calculate, as does LIS and all those other programs. I think PLDDT is in here too. Anyways, just no. What?
How do you graph off the JSON?
There are websites online that you can just drop this into and it will do the graph for you. I've had code at various times that will actually just use Python, graphing programs to make a graph of this program. You really want to use somebody else. It's not, I wouldn't try to, you could really go to ChatGPT or the Anthropic one, Claude, and just say, write me a program that takes this kind of file and makes a color-coded, a heat map file of that image.
Are you going to show the IPSA calculation from this file?
Yep. So let's hope this works. You can download IPSA. I'm not going to do it right now. It's just a Python script. And you use it on the command line. And you type Python 3, the IPSA.py, that's the name of it, the name of the JSON file, the name of the PDB file, and then 2 numbers, like the PAE cutoff and the distance cutoff for the context. So I do like 10 and 15. And then that automatically outputs the text files of the JSON file. Let me show you one other thing. And we'll hope this will. So a way to do it if you don't want to get involved in is this Livia server, Livia. The link is in the PowerPoint, but I'm going to click on that Livia server. This is by the group that does the LIS measure. But they include IPSA and other metrics. And so what it says here, it says drop prediction files or folders here. And so I can drop that zip file. Should put it at the top. Oh, that's where it is. Okay. Which was KRAS. So I'm going to click this and it says, select a file. And it was at the top. There's the zip. No, I need the zip file. There it is. So it's that zip file, this KRAS.zip. And hit open. See how quickly that did that? All I did was drop that file in there. You could do this with an AlphaFold 2 or an AlphaFold 3 zip file. And it's a really nice server. You get the name of the model here. You get the score between chain A and chain B. You get their ILIS score, their ILIA score. I think it's square angstroms or something. Here's the IPSA score. So for these 5 models, the IPSA scores are 0.822, 824, 816, 819, 791, and the average. Here's that active PTM. That was another model that we talked about. I showed you how the LIS score works a little bit. Here's the IPTM from the output of AlphaFold. So that's whatever AlphaFold.the IPTM. And you can download that as a CSV. Now you get a lot of stuff about their score because this is their webpage, so they're analyzing their score, which is totally fair. And so here are the scores between the chains. LIS, like IPSA, looks at each of the two matrix, this lower triangle between chain A and chain B, and the upper one between chain B and chain A. And they get different scores for those two pieces, so it gives you the two different scores. It doesn't do that for my IPSA score. Give you the PAE matrix, and then there's just lots of stuff related to their scoring system. It's really, really super useful, and probably the quickest way to, if you're only going to do it once or twice, getting the IPSA scores between your, and it will do all of the IPSA score, all the pairwise IPSA scores. For like if there are 3 chains, you'll get A to B, A to C, and B to C, all.
Did you need to set up an account for that, or is that just
Livia? You do not need to set up an account. You just go to this web page and drop it, and you're done. Yep, super easy. And there are various things you can, download. Explore the functionality, but it gives you all that information really quickly. And in fact, if I download the CSV and then open the CSV, you get, you actually get more scores that are not in the on the web page. Yeah, so super cool. Okay, so that was ColabFold. Now let's spend a little bit of time on the AlphaFold 3 server, which is here. So remember, AlphaFold 3 can do post-translational modifications. It can do DNA. It can do ligands. So when you go to the, I'm going to clear it. Clear button does not work.
Yeah.
Oh, it doesn't matter. So I have copied. So, oops. All right, I need to just get a new job. Okay. They introduced a bug to the web page recently. It's really annoying. Okay, so this is a cleared page. So this is what you get when you go on there. So the first thing says this is the first molecule. It's a protein. There's one copy. I can change this to multiple copies. So a homooligomer. So I'll put it back to 1. And then I just copy and paste a sequence in here. So if I go to this one, and I'm just going to copy the first sequence. So unlike ColabFold, where I had to put a colon between the 2 sequences, this one you don't. You put them in separately. So I'm just going to paste that sequence in. It doesn't want any other characters, any spaces, any colons. It will complain. You have to get rid of any other extraneous characters other than the 20 letters of amino acids. Over here, there's a button, three little dots, and one of them says PTMs. So if I hit that PTMs, the sequence pops up in a new window, and I can click a serine. There is a tyrosine in KRAS that's phosphorylated. It doesn't matter. Here's tyrosine 32. I'm going to phosphorylate. I just clicked on it, and it's residue 32. Now it says select PTM, and the only option it gives you is phosphotyrosine. And then you hit save. And it's yellow here, and it says phosphotyrosine residue 32 down here. You can do that as many times as you want and add as many phosphorylations as you want. You then add another entity, and I could add the second sequence in my PowerPoint, which is RAF1, copy, paste. There we go. Now the button says add entity. So I hit that, and now I got another box. I'm going to add a ligand. So I have to select protein and swipe down to ligand. And now there's a list of ligands that it will allow you to use. So there's only about 20 of them. I wish there were more. They really could put more. It would be a really benefit to science. But so I'm going to put in ATP. No, sorry, GTP. It doesn't make sense to do. There we go, GTP. There's also a magnesium ion bound to the active site of KRAS. So I'm going to add another entity, and I'm going to pick ion. And my choices are magnesium, zinc, chloride, whatever. I'm going to click on magnesium. Now if I hit, that's all I can do on the web page. Those are the only things I can change. Like just the input of the molecules. That's all you can do on the web page. You hit this button, continue and preview job. It gives you a random time up here. You really want to put a name for the job. KRAS _ RAF1 _ GTP. And then hit confirm. And then the job starts running. And so here are all the jobs that I've run previously, but here's that name job, and it is currently running. It won't email you or anything. You just have to watch it to see when it has stopped running. It will say done. I'm going to click on this one. This is a job that I ran previously of the same exact sort of job. And if I click on it, I get a page about the modeling that it did. It gives me a structure, color by PLDDT. Remember that the KRAS model? The helix was, I think I showed the colors the other way around. But this is the end that has poor PLDDT. Here's the PAE matrix. It tells you where the two chain boundaries are, which it didn't do before. And it gives you information. That's the thing. That's basically, and it gives you the IPTM at the top is 0.9, and the PTM of the whole structure is 0.9. That's all you get on there. And so and then you can, from here or from the list of jobs, you can download. And if I hit the download button, I get fold _ KRAS _ RAF1 gtpmg.zip. And that zip file should be up here. It's here on my desktop. I can go back to that Livia server and drop where did it go? Click the Livia server and just drag and drop that zip file onto the Livia server. Boom. And it gave me all of the scores. Now, I had two proteins, GTP and magnesium. So there's chain A, chain B, chain C, chain D. And I put them in that order. Whatever order I put them on the first page, in the input page, that's the order here, A, B, C, and D. So for instance, A and C is GTP, and A is KRAS. So I get really good scores from IPSA, from ILIS, from IPTM of KRAS with its GTP. Totally makes sense. A and B are the two proteins together. The scores are actually lower. But you take a look at the scores, and it gives you the average, which is sort of nice over the 5 models. A and D, are all zeros because A and D is the that's odd actually. That's the magnesium with the KRAS. That's strange. I wouldn't expect that. So Oh, I know why. I know why there's a, it cannot, there is no PAE involved, for an ion, for a single atom. There's no way to think about an aligned error, like what would it mean to align on a single ion sphere? So it doesn't calculate one. That's why those are all zeros. So the information is not available. So this is a very quick way to get that sort of information. In the last few minutes, I know the time is almost up. At least what you were, see, I lied to you and I told you we might get out early. But I do want to do Boltz, one of the Boltz servers. So this is ColabFold batch. If you run ColabFold on your own machine, you can specify all those things that were on that web page in this big long command line. This is on Smoothen, this is on Ben's project. There's AlphaFold 3. I put in some hints. If I open up that zip file, there's a file that has .json, and it's like summary confidences.json, has all of the pairwise IPTMs. And then there's a bigger JSON for every model that has the PAEs and everything else in it. Here's a link to that Livia webpage that we just looked at. But very quickly, I just wanted to show you a trick about Boltz-2, because you might want to do a small ligand that is not on the AlphaFold 3 server. Maybe it's a drug. Maybe it's, I don't know, PLP, some cofactor that just isn't a list on those 20. How are you going to do that? You're going to do that with Boltz-2. So there are several sites where you can do this one. I found this one particularly useful. You have to sign up for an account, but you get 30 free jobs a month or 50, I forget what the number is. And it's called TamarindBio. So here's the link. Tamarind, where are you? Where it's here. Tamarind. Okay. So these are jobs I ran. So this you have to sign in and go. You have to sign up for an account. So if you go to their main webpage, you see all of these different tools that you can run on their site. It's actually really amazing. So you could run OpenFold 3 on here. You can do protein design on here. I haven't tried it, but we're just going to hit the Boltz-2 button. And we're going to put in, all the same kind of information that we did on the AlphaFold 3 server. We're just now going to do it on this Boltz-2 server. So KRAS, RAF1, protein, I can put in the sequence. I'm just going to make and then if I hit the plus button, I can put in another sequence. So one of that's RAF1, I could put in KRAS here. And then I can hit the button again. And now, so it has that same kind of button that says protein, DNA, RNA, or ligand. So the trick I want to make sure you understand how to use is the ligand one. It gives you two different ways to put in a ligand. One of them is called the CCD code. So when you have a PDB structure, the chemical component dictionary, CCD, is the 3-letter code of the ligand. So GTP is just called GTP. Imatinib, I think, is SLI. Zanubrutinib is 012 or something. It's made that up. But so every small molecule ligand that is in the PDB has a 3-letter code. They ran out of 3-letter codes last year, and now there are 5-letter codes for some. So you have to think about the 5-letter codes. So I could just put in GTP here. And that's fine. And it even shows me a picture of GTP. But if I want to put in some drug that isn't in the PDB at all, I have to get something called the SMILE string. The SMILE string is just a series of letters, C, S, N, and O, carbon, hydrogen, for all the elements, bunch of parentheses on how atoms are connected, and other weird symbols. I don't really know how it works, but it's called the SMILE string, and you can write one down for any organic molecule. How are you going to get those? The easiest way, if the molecule is in the PDB, is to just go to the PDB website and search. And if I type GTP up here, GTP, the first thing that appears here, it says in chemical IDs. Everything below it is GTP, cytochrome, MOX, whatever. So I'm going to click that 3-letter code. because what I want is the web page on GTP, on the 3-letter code GTP. So there's pictures of it. I can rotate it around. But then there's this table down here that has the smile string. And so as I said, it's the letters, for the elements, but also parentheses and equals probably for double bonds. It specifies the connectivity and the valence of every bond in the small molecule. And all you have to do is copy one of these. I don't know if there's any advantage to one over another. So I'm just going to use the OpenEye canonical one, I think is the one I usually use, but not really any for good reason. So I'm just going to copy that. So I'll just hit Command C, and then I'm going to go back to the Tamarind web page. And so I had a choice, CCD code, I put in GTP. So I'm going to erase that. And I'm going to hit the smile string, and I'm going to put in that smile string. And actually, it drew GTP. It just interpreted that whole string and came up with that molecule. And then you just hit submit, and it runs the calculation for you. So you could put in any drug you want. If it's a drug that is not in the PDB, you can probably go to PubChem. Might be a site where lots of molecules are in there that are not in the PDB. You can search for your molecule by name, by structure on that website and get a smile string. If it's a molecule that's never been made before, I have no idea how you get a smile string. There are organic chemists probably would help you do that. I'm sure there's ways you could Google them. I don't know how to do it. So then you just hit the submit button, and I already ran this. So I ran this one, and so I hit the view button. And their view page is actually pretty good. There's a structure, I guess, colored by PLDT. Now it's just colored by chain. And it gives you a lot of the confidence metrics, including IPSA. Right, there's IPSA, and it gives you actually I think it gives you the minimum one. Remember, it's an asymmetric score, A to B and B to A. The default final one is the max, but it actually gives you both of them. So here's one of them, 0.84. Actually, those are all the same. I think one of these is the max and one of them is the default score, but does it give you the min? Anyway, it doesn't matter. It's got P.Q, it's got P.Q2. I mean, this one is pretty good you're running it, you can't do it with your AlphaFold thing and put it on here, but you can, if you run bolts, you get all this information. You get the heat maps for all of your, the PAE matrices for all of your models. You get the A3M sequencing. Remember the sequence alignment, that all these programs depend on. And then you get this, and I'm going to show you this quickly here. If you downloaded the zip file that has all this information in it, there'll be this file that will be something.json. And in AlphaFold 3, it's called something summary confidences something.json. And the reason I'm showing you is this. When you run AlphaFold 3 on the web page, on the server, you get one IPTM. What if your structure has 3 different proteins in it? What you really want to know is, what is the IPTM between chain A and chain B, between chain A and chain C, and between chain B and chain C? I want all of those, not just the one overall global IPTM. It's in this file, and it's in this file under this thing called pair chains IPTM. Pair chains IPTM. So what is it? It's a square matrix. It's basically every molecule in my input against every other molecule in my input. And it names the molecules in my input 0, 1, 2, and 3. So 0 is KRAS, 1 is RAF1, 2 is GTP, and 3 is magnesium, because that's the order I put them in. In my brain, that's the order, the way to construct this complex. Put the bigger molecules first and the smaller ones last. So for 0, the 0, 1, 2, and 3 is the IPTM of chain 0, KRAS, with itself, which is just the PTM of the one chain. It doesn't really make sense to IPTM of itself, but the second number, 0.861, is the IPTM between chain A and chain B, or the first protein and the second protein, KRAS and RAF1. And then the third number is KRAS and GTP, which is 0.86. And again, the ion is 0. Chain 1 here is RAF1. Binds well to KRAS, shouldn't bind well. It's actually strange that it binds that well to the, I don't understand that, to the GTP. Anyway, so for all 4 molecules, you get scores to the other, to the other 3 molecules. And you just have to think of it just a square matrix. So 0 with 0, 1, 2, 3, 1 with 0, 1, 2, 3, 2 with 0, 1, 2, 3, and 3 with 0, 1, 2, 3. So it is 5 o'clock. So I've shown you how to use these 3 programs on existing web servers. You can download the PowerPoint that I've shown you today, as well as the PDF. And there are hints and kind of instructions for all of those things that I have just told you out loud. But that way you'll have them in print. I'm very happy to, if you have projects that you run AlphaFold and you're puzzled about the results, or you want to know better ways to run it, I'm happy to do a Zoom session with you. Because I've looked at hundreds of these. And I know for certain protein classes of proteins, I know better ways to do things in other ways. I'm totally happy to do that, spend a half an hour with you on a Zoom call. And I'm happy to stay up here and answer questions for as long as you have questions. I don't know what time our dinner reservations are, but happy to do that for the next half an hour maybe. And you can always email me if you just have a simple question you want me to ask. We don't have to Zoom necessarily. I can answer a question for you. So thank you very much for this opportunity to do this. It's actually really fun for me, and this workshop has evolved over time. This is, I think, the best one I've given. Most complete and with more understanding. I gave this 2 years ago and didn't understand all these scores that well. So to compare that one to this one, where I think I have a much better understanding of how these things work, and hopefully I've shared that with you today, and I hope it helps when you use AlphaFold. So thank you very much for your attention.